Life's Early Earth, Chapter 20, Section 2. Learning Objectives. Explain scientists' hypotheses about the origin of life. Describe the theory that explains the origin of eukaryotic cells. Explain what fossils can reveal about ancient life. In 1953, chemists Stanley Miller and Harold Urey tried to test the hypotheses that organic compounds could have produced on early Earth. They warmed water in a sterile flask and added methane, ammonia, and hydrogen to simulate Earth's early atmosphere. They passed sparks of electricity through the gases to simulate lightning. The liquid circulated through their experimental apparatus for a week. The results were spectacular. Miller and Urey analyzed their results and revealed that 21 amino acids had been produced in their apparatus. A more recent analysis of one of Miller's experiments not only confirmed their results, but also found the experiment had produced an additional 2 amino acids for a total of 23. Miller and Urey's experiment suggested that organic compounds necessary for life could have arisen from simpler compounds on the primitive Earth. While Miller and Urey's hypotheses about the composition of early atmosphere were incorrect, more recent experiments using current ideas about the early atmosphere have validated their conclusions. Organic compounds could have been produced from early Earth. The discovery of organic compounds on meteors and comets has further confirmed these lab experiments. Several lines of evidence suggest that RNA rather than DNA was the first information-carrying molecule. The RNA world hypothesis proposes that RNA existed before DNA. From this simple RNA-based system, several steps could have led to today's DNA-directed protein synthesis. This hypothesis shown below is still being tested, but evidence make it credible. Recent experiments show that at least 2 RNA nucleotides could form in the absence of life. Other experiments shown how simple clay minerals can bind nucleotides which can polymerize to form RNA polynucleotides. In addition, certain RNA sequences are capable of catalyzing chemical reactions that include RNA self-replication. Eukaryotic cells, as you've learned, contain several kinds of complex cytoplasmic organelles, including lysosomes, endoplasmic reticula, cilia, and flagella. 2 other organelles, chloroplasts and mitochondria, are even more complicated. The evolution of these complex eukaryotic cells from much simpler prokaryotic cells set the stage for the rest of history of life. How did these cellular complexities evolve? The fossil record provides few clues to the evolution of cells. The oldest eukaryote fossils have been found in rocks 2.1 billion years old. Unfortunately, these microscopic fossils don't usually show details of internal cellular structure, so they provide few clues about early eukaryote evolution. The endosymbiotic theory, developed in the early 1960s, proposes that organelles in eukaryote cells were formed when different types of cells joined in a kind of merger. The idea was endosymbiosis, which literally means living together within. A great deal of evidence from molecular biology now supports the theory that many of the complex features of eukaryote cells evolved through endosymbiosis. The fact that some cells today contain bacteria and algae living as endosymbionts further supports this theory. Mitochondria, for example, are similar in size to bacteria, have their own DNA genomes, synthesize some of their own proteins, and are formed when pre-existing mitochondria divide inside dividing cells. For these and other reasons, most biologists now agree with the theory that mitochondria are the descendants of free-living bacteria that took up residence in the earliest eukaryotes. Similar evidence supports the endosymbiotic origin for chloroplasts. There are many different kinds of fossils. Fossils can be large and perfectly preserved as an entire animal, sometimes complete with internal organs, or they can be tiny as bacteria, embryos, or pollen grains. Many fossils are mere fragments or of an organism, teeth, pieces of jawbone, or bits of a leaf. However, sometimes an organism leaves only trace fossils, casts of footprints, burrows, or tracks, or even droppings. By comparing fossils to each other and to living organisms, paleontologists can propose and test evolutionary hypotheses. Fossils reveal information about the structures of ancient organisms, the sequential nature of groups in the fossil record, evolution of common ancestors, and the ecology of ancient organisms. Comparisons of body structures can be used to test hypotheses about the appearance, evolution, and the history of groups in the fossil record. Studies of evolutionary change in the body structures can also be used to test hypotheses about the evolution of living species, form common extinct ancestors, and the evolution of diversity.